Hey everyone, we're going to talk about meiosis today. So before we move on, why don't you take a minute, maybe pause the screen so you can take a look at our learning goals and success criteria. So think back to last lesson, we talked about mitosis where parent and daughter cells have the same number of chromosomes, and these chromosomes occur in two sets. These two sets are created, one set by the mom and the other set by the dad. And the total number of chromosomes, when they add up all them up together, is referred as the diploid number. Now, meiosis then is the reproduction that involves the union of two cells that forms a zygote. Now, that zygote contains chromosomes that are from both parents but does not contain double the amount of chromosomes found in your normal body cells. It is a special type of cell division that occurs only in reproductive organs called the gonads, and they make reproductive cells called gametes, which we're going to talk about later, which is the ovum or sperm. In meiosis, gametes are haploid in number. Haploid means they are cells that only contain one copy of each of the chromosome that the diploid parent contains. And here we can see that we're using N as our haploid, and diploid is 2N, which means there's double the amount, right? Because it's two of the pairs that the parents contain. And we talked about the two, which one comes from the mom, one's from the dad. Now, the first part of meiosis reduces the number of chromosomes from diploid to haploid, and we call that reduction division. So, for example, human sperm cells have 23 chromosomes compared to 46 chromosomes in the somatic cells. Now, each human gamete has 22 autosomes and one sex chromosome. So it all adds up to the 23, and the 22 will account for everything that's part of the body cells. And then the last pair is the XY or XX, which determines which gender it's going to be. So if you take a look here, we have the blue arrows that are going to be haploid numbers, and then the red arrows are diploid in numbers. You can see that the sperm and the egg or the ovum, the entire way, they're going to be haploid until fertilization. Once fertilization happens, their chromosomes will add up in numbers, which will create our diploid, which is 46, and it goes through mitosis and development. And then for the cells that are in the ovaries and the testes, which are the reproductive organs or the gonads, they will start creating these cells that will go through meiosis to create more ova and sperm cells. So the stages of meiosis. So the stages of meiosis occur in two major divisions, which is meiosis 1 and meiosis part 2. And each meiosis 1 and meiosis 2 all have prophase, metaphase, anaphase, and telophase. So in the purpose of our lesson, we're going to use names like prophase 1, prophase 2 to tell us which meiosis we're in. So you can see here that it starts with interphase. It goes through PMAT part 1, so prophase 1, metaphase part 1, anaphase 1, telophase 1. Then it goes into prophase 2, metaphase 2, anaphase 2, and telophase 2 at the end. So you can see how it's divided into meiosis 1 and meiosis 2. So we'll start off with interphase. It's very much like mitosis, where the chromosomes replicate before meiosis 1, and during that replication, chromosomes are not condensed, are not easily visible, and after the replication, each chromosome is made of a pair of sister chromatids paired by a centromere. Now for prophase 1, the homologous chromosomes form homologous pairs. So homologous chromosomes are similar chromosomes but not identical. They have the same genes but different forms of these specific genes. And when the homologous pairs come together, they'll form something called a tetrad. Tetrad means it has 4 to it, right? It's tetra means 4 in Greek. The homologous pairs will come from both parents, one pair from each parent, And during fertilization, the union of gametes form a diploid zygote. Therefore, all the somatic cells are diploid and are copies of the original zygote. And that's why each cell has one copy from mom, which is called maternal origin, and one copy from a dad, which is called paternal origin. So continuing on with prophase 1, during the pairing process, these chromatids will actually cross over. And the purpose of this is so that it allows the recombination of genes. These non-sister chromatids will exchange genes. So if you can take a look here, we have one pair that comes from one parent and the other pair that comes from the other parent. So let's say blue comes from mom and green comes from dad. What happens is one part of their genes will actually cross over and exchange. They'll switch places. Okay, so chromosomes will contain some genes from both origins, and this happens randomly. And as a result, every sperm and egg has a little bit of different variations of the gene. And that's why you and your siblings will look exactly alike. Because if this crossover does not happen, that means every chromosome from the genes that are coming together from the mom and the dad will look exactly the same. Now, without crossing over, every chromosome would be either paternal or maternal. So in metaphase 1, you can see that the spindle fibers are attaching to the centromere of each chromosome, and they're going to start to move both homologous pairs. All the homologous pairs are going to start moving to the equator. Now, which pair will go where will be totally random. So that means the majority maternal pair and the majority paternal pair can go on either side of the equator. So when they separate, it's going to be totally random on which cell has more genes from the mother and more genes from the father for that particular trait. We call that independent assortment. In anaphase 1, the homologous chromosomes separate and move towards opposite side of the cell and get pulled apart by the shortening of spindle fibers, very similar to our mitosis, except the difference here again is that they have the entire pair that's going with them. Now, telophase and cytokinesis does not necessarily occur in all cells, and if it doesn't occur, cell division goes directly to meiosis 2. If it does, homologous chromosomes begin to uncoil and spindle fibers disappear, and the cytoplasm is divided, nuclear membrane forms, and two cells are formed at the end. Very similar to mitosis here. Now, whether or not it occurs, that will be totally random. Okay, so each cell now contains one copy of the chromosome, two chromatids. So a second chromosome replication will not have to take place. And at the end of telophase 1, each cell contains some maternal and some paternal chromosomes. And each cell also contains mixed chromosome because of crossing over, combining maternal and paternal variation of genes. So as you can see here, the colors are a little different. You see here, this chromosome here has a little bit of the blue, majority red, and this big blue one has majority blue and some red in there. In meiosis 2, it is identical to mitosis. All the steps are pretty much the same, and each cell beginning meiosis 2 are haploid, but has duplicated chromosomes, and each chromosome now has two chromatids. Remember, there's going to be a little bit of variation in there. And at the end of meiosis 2, each cell are now going to be haploid, but contain unreplicated chromosomes. Daughter cells at the end are called gametes in animals, and daughter cells in plants are called gametes or spores. So let's quickly go through this. Prophase 2, nuclear membrane dissolves. The chromosomes become more visible and are still attached to the copy by the centromere, and the centrioles will move towards the poles of the cell, and then you have your lining up happening in metaphase 2. Spindle fibers will start to attach and they'll start to pull, which leads us to anaphase 2, where they'll break the centromere, pull apart sister chromatids, and each copy of the chromosome or each pair will go onto opposite sides of the pole. And telophase 2 is the last stage of meiosis, where the nuclear membrane starts to reform around the chromosomes, and the cell membranes start to pinch towards the equator, signaling the end of meiosis and the beginning of cytokinesis. And at the end, you have 4 non-identical haploid cells. And that non-identical haploid cell is due to the variation that we had during our crossing over. So we're going to just pause here because more will come in your gametogenesis lesson, which you're going to take a look at in a little bit.